Hi, for your lab 2, you're going to be looking at how to use a microscope. And so in the lab, it kind of gives you an overview of using a microscope. So make sure you go ahead and read the overview. You can see on the left side is the plant cell and on the right side is the animal cell. You can see that they're basically similar. There are some things that are different. Plant cells will have a cell wall. They also have a large central vacuole, which animal cells do not. You'll see that the animal cell has the cell membrane, which is basically a more flexible cell wall. The cell wall in the plant and the cell membrane, what makes them different is that the cell wall is a lot more rigid and gives the plant the structure like a skeleton does for us. So go through, take a look at the different parts, read through them so you can see what makes a plant cell different than an animal cell. In part A, you're going to read about how Stella is collecting pond water and how she's looking at it. It's going to talk about magnifications. The thing about magnifications in the first question is you're trying to think what magnification is the highest magnification in Stella's microscope. So at the end it says Stella's microscope has a 10x and then a 40x object lens. So you're going to have to multiply 10x by 40x, and then you'll get the highest magnification that Stella has. And then you'll click on it so that you can answer it. In the second question, you're going to be reading basically about how Stella is trying to get her microscope in focus and make sure that she's able to see it. So you're going to answer the question right here with the drop down with one of these questions of why maybe it's hard for her to see it. And so read through the scenario and answer the question. Watch this video for question 3. You're going to be taking a look at the slide that Stella's looking at. You're going to be looking at it and seeing what kind of things you can see within the microscope because it's at the 400x magnification. Watch the short 30 minutes video. Then you're going to read how she prepares slides. And so what you're noticing is you're trying to compare how the slide looks at 40x versus 100x versus 400x. What things can you see as the magnification gets larger? What do things look like as they are smaller? So you're basically going to answer in this box how you can tell the differences, what happens as the magnification goes up, and maybe what does everything look like in each of the magnifications. You can do comparisons to things that you maybe eat every day. But the main thing is that you're trying to see how magnification helps for you to kind of see the different structures. And when you look at the 100x and the 40x, you can start seeing some structures, and you may want to go back up to your plant cell and animal cell and take a look at structures that you can see and then add that to your answer right below. And question 4, you're going to see how Stella is viewing the microscope human blood cells. You're going to first analyze the cells using a wet mount. You're going to see how Stella sees it through the highest magnification. And a wet mount is basically putting a wet species or substance on the slide and then putting over a little cover and then focusing in so you can see. So watch the 17-minute video. Read right here about the human blood cells on the different slides. Talk about what you can and cannot see as in the magnifications. So 40x versus 100x versus 400x. What can you see? What can't you see as the magnification increases? What do you notice? Of course, you can always get any of the answers after you answer them. Look at the sample answer. If you see that you want to change your answer after looking at the sample answer, you can use the sample answer to help make sure that you cover everything. And then if you want to change it, you're going to want to retype your answer and then hit save and exit, because once you hit save and exit, it will save it, and then you'll have to come back to it and go back to the same spot that you were just at. So right here, it's really important. Be sure to identify at least one cell structure or parts of the red and white blood cells in your description. So when you're looking at here, you're trying to see what you can determine, what structure can you see in it. And normally you're going to look and it's going to be something in the middle that you can see, or maybe you can see the white blood cells versus the red blood cells as you look at the magnification. So then watch the video so you can see the two different plant cells that Stella is looking at through the microscope. Make sure you read all the descriptions, and what you're doing again is you're trying to determine what things are in the plant cells. So describe how Stella's view of these plant cells and their parts change as she transitions through the 3 levels of magnification. Be sure to identify at least one cell structure or part of the leaf in your description. So what can you see? What part of the cell, of the plant cell, really becomes apparent as you increase in the magnification? Based on the images taken through Stella's microscope, which cell structure could be clearly identified using a compound microscope? So what could you clearly see as you look through these? And then again, since she's looking at plants, I would definitely go back up to the plant cell, take a look at maybe what is on the outside of the plant cell, because you can definitely determine and see that within the magnification of the compound microscope. Based on your images, sorry, I was just rereading the same question. Then right here, what you're really looking at is you're trying to see the difference between the cardiac muscle cell as you increase, how you can kind of see the fibers of it. What do they look like as you increase the magnifications, how the fibers basically bundled together. And then you have a piece of a frog skin. And you look at the frog skin and you can see how they're different shape, and the way that they're compactly together, you can see that they're different. One of the things you're going to want to realize is that the structure or the shape of a cell really helps determine its function. So in here, you're trying to really determine how the shape of the cardiac muscle, and think about what a heart does, versus the skin cell and what skin cells would do. And why would they be shaped differently? You can definitely notice there's a lot more cardiac muscles. They seem really tightly compact. So think about what the heart's doing, versus right here, they are compact together, but they're not as tightly compact. What are they trying to do? What does what does our skin cells do for us? How do they help us? What is their function? Because plants don't have interior or exterior skeleton or exterior skeletons, they require the cell walls to give them structure. Did you see any evidence of the cell structure in the plants? How is it different than animal cells, blood cells, cardiac muscle cells, and skin cells? So again, give your best understanding, explain as much detail as you can about how the cell wall helps a plant cell versus how it's different than animal cells like blood cells, cardiac muscle cells, and skin cells. The main thing is you just try to explain as much as you can. Again, you can use a sample answer to check your answer. Do not copy it word for word, but you can use it to expand it. So again, you're asked another question. Do the best you can on these questions. The main thing I'm looking at is to see how much you explain, how much you try your effort into it. I'm not looking for the perfect answer, but I'm looking for the effort that you're putting into it. One of the key concepts of this lab is so that you can understand how a microscope helps, and also secondarily how cells are the building blocks of life, and how basically cells work together, and the way that they function could be dependent on their shape and what type of cell they are. Question number 6, I want to just touch really quickly. It's looking at an electron microscope. Electron microscopes definitely let you see a lot more details because they have a system set up so that you can see smaller particles and the magnification is exponentially greater. So the reason why you look at a electron microscope is to see very small particles such as atoms, and so this allows you to see it more up close. So of course, when I ask you is what do you think electron microscopes allow a greater magnification, or why do you think electron microscopes allow a greater magnification than a compound? You're going to have to read the description and give your best guess. Again, you can use the sample answer to help you answer it, but make sure you do not copy it word for word. And then you have your last question. You're going to go ahead and read how this image of white blood cells you saw in Stella's compound microscope, what are the pros and cons of using an electron microscope in your response? So right here you have an electron microscope view of the white blood cells. You can go back up and compare it to the view you have from Stella's compound microscope. You can see that you can definitely see a more detailed image from the electron microscope, and you just have to explain how that can be more beneficial than using a compound. Once you're done with that, make sure you hit save and exit. Make sure you click all the way through to number 8 and then hit save and exit, and then it should turn it in automatically for you. If you have any questions, please feel free to contact me through email, text me, or call me on my number. Have a great day.